Hello everyone, welcome back again. So today I have come up with a video where I'm going to discuss a detailed explanation of CSIR Net Life Sciences answer key, that is 2022. Here I'm going to discuss the morning shift of cellular biology, that is unit 2. So all the questions of part B and part C I'm going to discuss here. So let's get started with the video. Few organelles that are present in a eukaryotic cell are mentioned below. So here are some organelles that are present in eukaryotic cell. The first is centrosome, second peroxisome, third is nucleolus, and fourth is endosome. Here they are asking which one of the following options represent organelles that are not membrane bound. So here you have to select organelles that are not membranous in structure. So if you see, centrosome is a structure that is without a membrane and it is associated with cell division. But this structure is without membrane that encompasses centrioles, and centrioles are only involved in cell division. But this is not bound with any membrane. Second is peroxisome. Peroxisome is a single membrane bound organelle that is involved in a cellular metabolism, or you can say oxidation of fatty acid. But this is a single membrane bound structure. So this option goes wrong. This is right. Third is nucleolus. Nucleolus is a structure that is present in an organelle, that is nucleolus. And what is the main function of nucleolus? It is responsible for the ribosome biogenesis. But this structure, if you see inside the nucleus, this is without a membrane. So again, this is without a membrane. So this option goes right according to the question that is asked here. The fourth is endosome. If you see the endosome, it is a structure that is responsible for intracellular sorting mechanism, or you can say it is involved in the transport of proteins as well as lipids among all the subcellular compartments. Now let's see the question what it is asked. Which one of the following options represent organelles that are not membrane bound? So here we have seen centrosomes and nucleolus are the organelles that are not membrane bound, because peroxisome has a single membrane and endosomes also has membrane, and we know that there are two types of endosome, early endosome as well as late endosome, that turns from lysosomes, that develops into lysosome. But both these structure, they have membrane and also lysosomes have. So accordingly, the option A and C is the right answer. So where you can find option A and C, it's there in the third option. So this option goes right. So let's see the explanation for this answer. The centrosome is a non-membranous nucleus-associated organelle that is associated with the nucleus and is involved in cell division. Whereas nucleolus is a dynamic membraneless structure whose primary function is ribosomal rRNA formation, or you can say ribosome biogenesis. So here you can see this is a structure for centrosome, and you can see the membrane that is disintegrated, means it is non-membranous structure. And also nucleolus, it is a non-membranous structure that is present in the nucleolus. So the question number 2 is about individual chromosomes are clearly seen during which phase of the cell cycle. So this is the very direct question that is asked, where you have to select key chromosomes. If you want to visualize, it will be visible during which phase of the cell cycle. Is it G0, it is G1, is it S, or is it M? So if you know the phases of cell cycle is divided broadly into two phases. It is interphase as well as the division phase, or you can say the mitotic phase. This division phase is called as the M phase, but the interphase is divided into G1, S as well as G2 phase. And before this G1 is a G0 phase where it is called as the resting phase. It is a resting phase for the cell where the cell enters in the quiescent state. So you cannot see any chromosome here. So this option goes wrong. What about G1 phase? This G1 phase is the preparatory phase for the S phase, because all the DNA synthesis or any copying mechanism, or you can say amplification of DNA, is taking place in the synthesis phase or S phase. So all the enzymes or protein that are needed for the DNA synthesis, they are synthesized in G1 phase only. So you cannot see any chromosomes here because chromosome is about to duplicate, or you can say DNA is about to duplicate. What about S phase? S phase is the duplication phase where chromosome will get copied, where DNA will get copied. What about G2 phase? G2 phase is a preparatory phase for the mitotic phase. Now what happens in mitotic phase? This mitotic phase is categorized into prophase, metaphase, anaphase and telophase. So if you see the diagram for all these stages, so interphase consists of G1, S as well as G2 phase. So coming to the prophase, mitotic phase, metaphase, anaphase and telophase. So let's see. In prophase, there will be a chromatin condensation and centriole migration towards the opposite fold. But you can see the chromosomes are not aligned. But as the cell division or as the cell enters from prophase to metaphase, there will be a transition and it enters a pro-metaphasic state. There happens a disintegration of nuclear envelope and also the spindle formation. After this, the cell enters in a late metaphasic state, which is called as the actual metaphase state. And you can see the nuclear envelope is disintegrated, means there is no nuclear envelope. And all the chromosomes are aligned in the center and the spindle is attached to the chromosome. So the chromosome will be beautifully, you can say it will be visualized under the microscope because the nuclear membrane is not there, it is disintegrated in the pro-metaphasic state only. And also the spindle is attached to the chromosome, the structure that is aligned in the center part. So in the metaphase, you can see the chromosome structure well. So this metaphase is included in M phase itself. So the right answer will be M phase, that is option 4. So here the right answer will be option D. The eukaryotic cell cycle is traditionally divided into 4 sequential phases as we have seen, G1 as G2 that are interphase and M is the mitotic phase. G1 as G2 together they are called as interphase. Chromosome duplication occurs during the S phase of the cell cycle where most cell components are duplicated continuously throughout the cycle. During the M phase, the replicated chromosomes are segregated into nuclei or you can say and that will be followed by cytokinesis. So mitotic phase, firstly karyokinesis will take place and then cytokinesis. That's why chromosomes are visualizing or it will be better seen in case of M phase when the cell is in M phase of the cell cycle. So this is about the explanation for question 2. So let's come to the last question of part B, that is question 3 in unit 2. Which one of the following conditions represents auto-polyploidy? Now again this is a direct question. So here if you see auto stands for self, poly stands for many and ploidy stands for fold. Auto means self, poly means many. Poly means many and ploidy means fold. Means many fold of something of same species is present. Self means same species. So if I am considering myself, so I am a single species. So same species ka many folds are present. But what many folds? Many folds of chromosomes are present. So let's see the options here. So option A is talking about more than 2 sets of chromosome, both are which from the same parental species. Abhi tak ye option looks fine. Second option is more than 2 sets of chromosome. Why more than? Because more than 2 set will come under poly itself. So both the options seems correct. But here in the next statement, that means in B statement, it is talking about both of which are coming from the different parental species. This is not the different parental species. This will be the same parental species because it is talking about auto. Auto means self. That means this A option means it is representing the auto-polyploidy condition. But what about option B or option statement B, that is talking about different parental species? Allo means different, means this condition is for allopolyploidy. What about option 3? More than 2 sets of chromosome, correct? Only from a single parent, not only both of which are from the same parental species. We have to consider at the species level. So from here, the first option goes right for the question that is asked for autopolyploidy. So the right answer is the option 1, that is option A. And last statement, if you see, it is talking about duplication of a chromosomal locus leading to spontaneous increase in the copy number of a gene. It is not the spontaneous increase and it's not about duplication. So with this, option first is the right answer. So autopolyploidy appears when an individual has more than 2 sets of chromosome, both of which derived from the same parental species, not different. And if it is talking about autopolyploidy, then there it will mention different parental species, and there you have to select the correct option. That is different. But according to the question, option 1 is the right answer. Now let's come to part C questions. So we have discussed 3 questions from part B. So the first part C question is, following statements were made about the cell cycle regulation in eukaryotes. So here you have to select the correct statement for cell cycle regulation, how it is regulated in case of eukaryotes. So the first statement is activity of maturation promoting factor rises and falls in synchrony with the concentration of cyclin B. So let's come to the diagram for this statement number 1. So here you have to select the combination of the correct statement. So here you have to select the combination of the correct statement. So let's go for statement first. So here if you see the maturation promoting factor, how it is formed? Maturation promoting factor is there for all different stages of cell cycle, whether it is G1 phase, it is S phase, G2 phase or M phase. So different type of cyclin and CDKs, they are assembled together and they form maturation promoting factor. Like for G1 phase, it is CDK4/6 along with cyclin D. For S phase, it is CDK2. For G2 phase, again it is CDK2. For M phase, again it is CDK1. And for G1 phase, the cyclin is D. For S phase, the cyclin is E. For G2, it is A. For M phase, it is B. So both these cyclin and CDKs, they are associated together and they form a maturation promoting factor. Means a factor that is promoting the maturation of the cell, or it is proceeding the cell cycle in a forward direction. So it is talking about activity of maturation promoting factor rises and fall. So every time what happens? If the cell is in G1 phase, so during G1 phase, this cyclin and CDKs are needed. But when the cell is transitioning from G1 to S phase, so it requires different set of CDKs and cyclin, that is called as 2E. So here what happens? All these CDKs and cyclin for G1 phase, they are degraded because now the cycle is entering into the S phase. And this 2E part, it is responsible for G1 to S transition. Now once G1 to S transition is completed and the cell has entered the S phase of the cell cycle, means now it's a time for cell division, or you can say the DNA synthesis. So DNA synthesis on maintaining the S phase of the cell cycle, it is done by CDK2 and cyclin A, and this time 2 and E will be degraded and 2A will be there because it has to maintain the S phase of the cell cycle. And now if it is done, S phase is completed and cell reaches to the G2 phase. Now again, if there will be a transition for G2 to M phase, there will be a new set of cyclin and CDK, and this time the cyclin and CDK will be CDK1 along with cyclin B. So here this time 2 and A will be degraded because here they have to maintain the concentration of M phase cyclin, which will cause the entry of the cell cycle from G2 to the M phase. So let's come back. Activity of maturation promoting factor rises and fall in synchrony. मतलब जैसा जैसा cell cycle होगा, accordingly there will be a rise and fall in the CDK and cyclin level with the concentration of cyclin B. It is correct. It is talking about cyclin B, yes, but this happens in case of cell cycle regulation. So this option is the right answer. So where you can see option A, you can see option A only in two options, that is option 1 as well as option 2. So here this option goes wrong. So let's read the second statement. CDC25 phosphatase mediates the removal of phosphate from the inhibitory tyrosine residue to yield highly active maturation promoting factor. So what happens when the CDK1 is there? So this MCDK is still inactive. How this will be activated? Because once this will be activated, this will bind to cyclin and it will form a maturation promoting factor. But you can see CDK, they are still inactivated. Because now this CDK inactivated, they will be acted upon by a V1 protein. Now, what is the function of V1? It will add a phosphate group to the tyrosine residue at 15th position. What is the function of V1? V1 add a phosphate group to the tyrosine residue of the 15th position and make sure this phosphate group is an inhibitory phosphate that inhibits something. So this phosphate group is added by V1. So let's talk about CAK, CDK activating kinase. You can only see kinase that activates the CDK, means this CDK will be activated. How? Because this CAK, that is CDK activating kinase, will add a phosphate group to the threonine position at 161. So at threonine ka 161 position, one phosphate group will be activated and this makes the CDK active. But fully CDK will not be activated. Why? Because this is the activatory phosphate that is added by CAK. But you can see an inhibitory phosphate is added by V1 that doesn't make MCDK fully active. So what happens in the next step? So here you can see the inhibitory phosphate is there that is added by V1 and activating phosphate at threonine 161 position, it is activated by or added by CAK. In the next step, CDC25 phosphatase will come. And what is the function of phosphatase? To remove any phosphate group. So it will remove the phosphate group from the tyrosine 15 position because this was added by V1 and that was an inhibitory phosphate. And now the MCDK will be fully activated. And this is how the activation of cyclins and CDKs are taking place and that will form a maturation promoting factor. So CDC25 phosphatase mediates the removal of phosphate. Yes, it is a phosphatase that will remove phosphate from the inhibitory tyrosine residue Y15 that was added by V1 protein so as to yield the highly active MPF. So again, this option is the right answer, that is option B. So this is the correct statement. So here you can find option B in both the option, that is option 1 as well as option 2. So now let's read third statement. So MPF specifically phosphorylate and depolymerizes lamin A and C but not lamin B. So this MPF, it is specific for lamin A and B2. It is specific for lamin A and B2, but here it is saying it is not specific for lamin B. So this option goes wrong. So where you can see option C, it is there in the first option. So accordingly, this option 2 is our right answer. So MPF, when it is activated, it will phosphorylate a set of protein that includes H1 histone, it includes MAP protein, it includes centrosomal protein that is involved in cell division and also the intermediate filaments of nucleus that is called as nuclear lamins. So this option is correct, that is option D. So this option is also correct. In Schizosaccharomyces pombe, overproduction of V1 protein decreases the length of G2 phase. So whenever there will be an overproduction of V1 protein, now you only think if V1 protein will be more, then it will extend the G2 phase because inhibitory phosphate is there. Because V1 is overproduced, it will always add an inhibitory phosphate to the tyrosine 15 position. Means it will increase the length of the G2 phase and it will decrease the period for the M phase. because M phase period will be decreased because CDK and cyclins, they will not become activated and they will not form maturation promoting factor. So there will be no transition from the G2 to M phase of the cell cycle. So if there will be an overproduction of V1 protein, the cell cycle will not proceed and the length of the G2 phase of the cell cycle will be more and it will decreases the period of the M phase. So this option is contradictory. So this option goes wrong. So accordingly, option 2 is the right answer. So here we have seen the diagram and also we have seen the explanation for this. So now let's come to the next question, or you can say the last question for this unit 2. That is, you have to match the CDKs and cyclin. Again, you can see there are two questions that are asked from cell cycle and division. And also in part B, we have seen the chromosomal stage that was asked in which phase of the cell cycle chromosomes will be clearly visible. So you can see cell cycle is the main part from where questions can be maximum asked. So again, here you have to match the column like enzyme and it is asking for cell cycle functions. So again, we'll come to the diagram and we'll see first. So here whatever I have explained in the next question is asked here itself. So G1 phase is maintained by cyclin D along with CDK4 and 6. So this arrow is for maintaining the cell in the G1 phase. What about if the cell is taking the transition from G1 to S phase? That will be maintained by cyclin E along with CDK2. But when the cell is entering directly into the S phase and it is regulating the DNA synthesis phase, that time it will be regulated by cyclin A along with CDK2. What about cyclin B or A along with CDK1? That is taking care for the transition of G2 protein to M phase and maintaining the M phase. So this is about the cyclins and CDKs that are involved. So here we have seen CDK1 is responsible for the mitotic entry. It is causing the transition from G2 to M phase, and CDK2, they are involved in G1 to S transition, or you can say DNA phase ka synthesis. Now let's come back to the question and see the right answer. So CDK1. So CDK1 is asked for cytokinesis, S phase entry, mitotic entry or Anaphase onset. So just now we have seen CDK1, it is the last CDK that is acting, that is responsible for mitotic entry. So where you can see the mitotic entry in third option. So A matches with third. Where you can see A matching with third? Only in one option. So this option goes right. So now let's come to the next option and see whether they are right or wrong. So the next option is APC-C CDC20. This APC is responsible for the transition of metaphase to anaphase. Abhi tak we have seen CDK is responsible for entering the cell into the M phase. But what about M phase of cell cycle? M phase has prophase, metaphase, anaphase and telophase. But when the cell is entering from metaphase to anaphase, this is done by a separate set of protein that is called as APC-CDC20. So APC-CDC20, it is for the anaphase onset, or you can say the metaphase to anaphase transition. So B matches with 4. Yes, B matches with 4. CDK2, we have seen it is matching with the GS1 to S transition. So it is causing GS1 to S transition, or it is also responsible for S phase ka cell cycle. So CDK2 matches with S phase entry. So C matches with 2. And Aurora B is a protein, or you can say it is an enzyme. You can see in the diagram as well, in the above diagram. Here it is Aurora B. So the new karyokinesis is completed, you can see, and you can see the cleavage furrow here. That means the cytokinesis is about to happen. Means karyokinesis is completed and cytokinesis is happening, and this furrow is formed. This furrow later it will divide into a 2 daughter cell. You can see 2 different daughter cells are formed. And who is mediating this cytokinesis after karyokinesis? It is Aurora B enzyme. So D matches with first option. So here D matches with first option. So the right answer for question number 5th is option 2, and this is the right answer. So now we have seen the explanation for all the questions that was asked from cellular biology, that is unit 2 of CSIR Net Life Sciences, where we have seen 3 questions from part B and 2 questions for Part C. So now I have come to the end of my session. I hope the discussion was helpful for you. If you find it helpful, please do hit the like button and subscribe to our channel, that is Biotecnika. Thank you for watching this video. Meet you in the next session. Till then, bye-bye. Take care. Have a nice day.